welcome back after snow day last week. Everybody did all right, I suppose. And everybody did get the video to watch it. It went okay. All right, very good. All right, so let's look at what's coming up here. Last week was, I think these dates are incorrect, right? Today's the 23rd, so. But the order is correct. So last time we started learning about bacteria, kingdom Monera, okay? And we're going to finish that today. Now look what happens in Module 10. I spend 3 weeks on fungi because there's enough stuff in there and it's really interesting. Not to mention in this first part right here, I'm going to talk more about microbiology, okay, because I just can't help myself. You're going to be taking home today a little petri dish and you're going to use it to culture something at home. And so I'd like to cover that a little bit at the beginning of next week's class, and then we'll start learning about the fungi. All right, then we're going to move on to 11A and B. Look how close we are to spring break, right? About 6 weeks away. How about that? And then after that,
a whole week?
Well, we just don't have any classes that week, so yes, it is a whole week.
After spring break, will we start the dissecting part of biology?
We're going to, technically, we'll be dissecting here in fungi, and we'll be dissecting flowers. We'll be dissecting plants, if you will.
They're just
but the creatures we're going to dissect. Yeah, I got all the frogs in. They all arrived.
All the frogs and wolves?
They still need to find a baby alligator somehow.
Yeah.
Yeah, I don't have that, so.
Need to get one somehow. Start calling around the zoos.
Can I bring a rabbit?
A live rabbit?
No.
No. I don't want to have to kill anything.
No. If I kill it, can I bring it?
I wouldn't be happy with a fresh sample because, strangely enough, the preserved ones can't harm us in any way, but fresh samples can. They could have bacteria growing on them.
All right,
Which we're talking about bacteria right here.
So, like, kill it and then put it in the freezer for a couple days.
But then we have to thaw it, you know.
Then
So.
thaw it out.
I did that with the
Toss it
shark.
in
Remember
the
I told you I
microwave.
found the dead shark on the beach that time and I came home, put it in the freezer, and we used it for several years. I kept putting it back in the freezer, but then finally it was just so stinky that I had to get rid of it. Okay. Here we go. Kingdom Monera continued. Remember, Monera is sometimes considered an antiquated term. They're putting that into the eubacteria and the archaebacteria. I should have talked about that last time. Does that sound familiar?
Yes.
Yes? Okay. Y'all were able to fill in your blanks from part A? No problem? Okay, good. All right, so let's look at bacteria now a little more closely. The eating habits of bacteria. Here we go. Prokaryotic organisms must have a way to get energy from their surroundings, like everything else, right? Remember what a prokaryote is? I think probably last time you had to draw a picture of it, yes? That sound right?
Sure.
Prokaryote means they don't have a nucleus. They don't have any membrane-bound organelles. Their DNA is just floating around inside of them. And that's bacteria. Okay, so that's the case there. Most bacteria are decomposers, also known as saprophytes. So saprophyte is an animal that eats dead stuff to survive. Okay, that's their food source. Some bacteria are autotrophic. What does that mean?
That means they make their own.
Good.
energy.
They make their own food, right? In other words, they're photosynthetic. We'll look at a couple of ways, but the most famous would be something called blue-green algae, which is actually a bacterium called cyanobacteria, one of the most important oxygen producers on the planet. The process they use to make their own food is either photosynthesis, in the case of blue-green algae or cyanobacteria, or there are some really crazy ones at the bottom of the ocean using something called chemosynthesis. Instead of the energy from the sun to go through metabolism, they use the energy from chemicals. And typically the chemicals
Don't they get
are...
those from plankton or whatever?
It actually is it's seeping out of deep ocean crevices where the heat kind of comes up and some chemicals are released. So it's happening usually down deep in the ocean. But it could happen in other areas where chemicals are available for that kind of thing. All right, so just like anything else, they have to get their energy from somewhere. Here's a chart. Do you have a chart in your book?
Yes,
We
ma'am.
do.
Here we go. Diverse ways that bacteria eat, okay? So either they're autotrophic. Do you have these things at the top listed like I have? Yes?
Yes, ma'am.
Okay, good. So they're either autotrophic or heterotrophic, one or the other. So they can be photoautotrophic, meaning they use the sun's energy, so photosynthesis, or chemoautotrophic, they use the energy from chemicals. The source would be sunlight in this case, and inorganic chemicals in the second case. Carbon source would be CO2. Remember the photosynthesis equation. Question?
Does chemoautotrophic have anything to do with like chemotherapy?
Well, chemo means chemicals.
Okay.
So chemotherapy, chemical therapy. So
Okay.
yes, yeah. So the base word is similar there. Our heterotrophs are either photoheterotrophic, using sunlight or stuff they get from the sunlight. Chemoheterotrophic, they use organic compounds. Saprophytes mostly, that means they're eating dead stuff. Some of them are parasites. Parasites, remember, often can cause harm to the host and when they're in parasitic symbiosis. And then they use organic compounds as their carbon source. So bacteria are considered simple life forms, and it's pretty amazing how complex they really are, actually. They're really simple about that life. Here we go. Oxygen. As we know, for us, we require oxygen to survive, right? A lot of bacteria do as well. If they do need oxygen to survive, like we do, they're called aerobes. You've heard of aerobic respiration. We go through aerobic respiration, meaning that we need oxygen in order to survive, okay? But not all bacteria need oxygen, okay? So if they're obligate anaerobe means they must have oxygen to survive. We, human beings, are obligate anaerobes. If we're put in a position where there's no oxygen, we will die, all right? Not all bacteria are that way, however, okay?
Is that like the like crabs?
Crabs.
I think crabs still need oxygen. You talking about the creature?
Well, like aren't there like certain crabs who can live underwater for
Sure.
long times?
Yeah, we have land crabs and we have sea crabs. So the sea crabs breathe through gills. Land crabs breathe through lungs. Yeah, but each of them can be, a little bit on land, a little bit in the sea, kind of a thing. But they have to go back to their regular spot to get oxygen. Yes. Question? No. Okay. Obligate anaerobes. This is interesting. The obligate anaerobes. That means they are obligated, or they must have no oxygen. Okay, they do not require oxygen for cellular respiration, and in many cases, almost all the cases where they're obligate anaerobes, they die in the presence of oxygen. Isn't that weird to think of it that way? The oxygen kills them. All right, so some of the really bad ones are in this group, some of the really bad pathogens. All right, so they say, no, we don't want any oxygen. If you're going to work with these in a laboratory, you have to have a way to work with them without letting any oxygen into the system.
Like a vacuum.
So
cleaner.
it's tough to do, right? It's tough to work with these guys. You can put them in a pouch and with a little oxygen absorber in the pouch, like almost those things like you see in your food that dries things out. It's similar to that, but it actually literally takes the oxygen out and you can like a vacuum suction on a bag or something, but they have to stay in the bag the whole time while they're growing because they can't have any oxygen. All right, some bacteria can form something called an endospore. It's a thick internal wall. That's this little round circle thing you can see within this oval shape right here. Produced by the bacterium and encloses the DNA and other essential parts. So what can happen at this point in time is that if things get tough, because the number one enemy of bacteria, did we talk about it last time in the video? The number one enemy of bacteria is drying out. Mm-hmm. Okay, yeah. Desiccation is the word. And so in this particular case, this type of organism can, in a time when things are getting dried out, they can take their DNA and wrap it up in this special endospore, and it kind of can go through a dried out period, and when things get wet again, it'll regrow. So it's a way of kind of sort of going dormant. Mm-hmm. Okay, so they'll do this. A lot of the bad ones, the pathogenic ones, are in this group as well. All right, because they'll take their DNA and other essential things and just lock it up inside that little spore. And you can see right here, what they'll do is they'll go through stages when they realize that it's starting to get dried out or some other stress is on the system. You can see how they're going to move all the DNA over to an edge right here, form that little spore around it, and then there's a hard case on it over here. In fact, it's such a hard case that sometimes when you see it, it sort of looks like a powder. Okay, which brings us to one of the most famous, or rather infamous, examples of this, and that is anthrax. You've heard of anthrax?
Sounds familiar. I think you mentioned it in the last
In the video?
video.
Okay, very cool. All right, so there's two forms of anthrax. One of them is very deadly. That's the one on the right over here, the one that invades your lungs. We also can have intestinal anthrax symptoms that cause really bad gastrointestinal upset. It's bad, it's not good, but it doesn't typically kill you. The one, though, that gets into your lungs does. Now, think about this for a second. If we have it's a bacteria just living, right? And if it gets into, like, say, a creature, animal eats contaminated grass, and that grass has anthrax growing on it, and they'll get this over here on the left, this intestinal problem, and they'll be sick, and maybe we can use an antibiotic or something like that. Now, if this anthrax has been stressed out and it's produced that little endospore, it can actually be in sort of a powdery form. And think about that. Think about a powder that gets into the air. Will I breathe that into my lungs?
Mm-hmm.
I can breathe that into my lungs, and think about the inside. My lungs are very moist. It's important for our lungs to keep moist inside. We don't want them to be all dried out. We don't want them too moist because then we'll drown. So guess what? That moisture wakes up that bacteria. That endospore bursts out, starts producing and reproducing all this stuff inside your lungs, and you die.
What can anthrax grow on? Is it like mostly on food and stuff?
Tissue, living tissue. Yeah, but where do we find it? Of course, it grows like in the dirt, in the soil. You can find it in the soil. Now, y'all aren't going to remember this, you're too young, but there was a attack on our country back in 2001 called 9/11. Yeah, you've heard of it?
Yes.
There was, very interestingly, of course it was terrible, right? Planes flew into buildings, it was awful. We all saw it happen on TV, and we're like, what is happening? Almost simultaneously, there were people around the country getting envelopes in the mail with a white powdery substance, and it was anthrax. So we thought, what is going on? We've got anthrax attacks, we've got planes flying into buildings, it's nuts. Well, it turns out, I think it took several years to find out it was some scientist who sent that out just to kind of create chaos. Nobody got sick from it. I know, he got arrested. He was, I think he's in jail right now for having done that. That's bioterrorism, they would call that, if you're using biology to hurt somebody. I would call COVID bioterrorism personally.
Yeah, for sure.
And they're talking about, disease X, watch out for disease. I don't know if you've heard of that disease X coming around. It's like the next planned situation. Yes?
Are there still like biological weapons?
Absolutely. You betcha.
I thought those weren't real.
They're not legal,
Oh.
but they are definitely real.
Could you like bioengineer a person to where you can make them stronger, have like certain things?
I would suspect so. I don't know the mechanism for it, but yeah, I think some stuff is coming out. This whole like bioengineering stuff's coming out. That's pretty wild stuff, pretty crazy stuff.
So hypothetically,
You could be the next Captain America.
hypothetically, if I was from another country and was sent by my government to another country
Sound a little suspicious
to
there, Eddie.
strangle a man, would that be considered bioterrorism?
I think it would be considered murder. I don't know that it's bioterrorism.
But I'm a biological
I mean,
species.
I mean, I
if
guess
you're getting
technically you
technical
could say
with
that.
it, you could, but
Typically, bioterrorism has to do with using a biological agent of some
So-
sort. In your case, I guess your hands, where you're the biological agent. Yeah.
would be like pipe bombs be qualified under biological stuff because they're using
It depends on
different
what's in it. It could,
chemicals and stuff?
because there's chemical.
Yeah,
If you use chemicals to blow something up,
interesting.
then that would be chemical terrorism. Yeah.
Back in the 1920s, whenever girls would paint the watches with like radium paint,
Watches?
watches like clocks and watches with radium paint,
Also
it glowed
use
in
that
the
for
dark?
toothpaste.
and like and they had to lick the paint, and they were told to lick the paint. Is that technically
That's probably chemo.
bioterrorism.
Because that's a chemical. Right.
Yeah,
Weird things.
but they were telling them
What is the story? Where'd you hear about this story?
like radium girls.
Really?
Yeah, it like history thing.
Interesting. Because, yeah, that'll make you sick.
Yeah.
It takes time, too. It's not like a
Weird
quick thing.
thing. They were
Yeah,
using
and they were
that
just,
to
like,
make
keep on
toothpaste at one point.
Radium?
Yeah.
To make toothpaste? So
Yeah.
your teeth will glow?
They also did it for medicine.
That was the whole reason why
I don't
it got
know this
bought.
story.
Imagine like going on a
People
walk
would
in
also
the
make
night.
like
And seeing just a
magical
blue
potions
smile
out
coming
of
at you
it.
from the woods, some horror movie
Because it
stuff.
glowed?
They're like some
It was like some kind
glowing
of they would
contacts.
like say it's like this elixir that cures everything.
Instead, it killed everything.
Windex.
Yeah.
They died.
Cheers.
Wow.
They
Okay.
took it and they died.
Yeah.
Okay, well,
Wait, does that qualify
would
as
be-
bioterrorism?
well, I think that would be chemo.
Listen.
It'd
Or
be
is that-
chemicals.
Is that accidental chemoterrorism?
Or stupidity, maybe? Just
That
I
could
think the
be.
stupidity is
Yeah.
that.
Yeah. So. All right, fermentation. So if something's a facultative anaerobe, facultative means it can survive in no oxygen. It prefers to live in oxygen, but it can actually survive in no oxygen. Okay, but so they can use O2 when it's present for respiration, but when it's not present, they will go through something called fermentation. And you've probably heard of fermentation. It's the very same thing, the fermentative process that causes
Sourdough, right?
sourdough bread, beer and wine, and vinegar, and those kind of things. Yeah.
I think you told us a bit about it when we were doing that iodine
It was
cornstarch experiment.
well, it was probably when we were talking about Krebs cycle and everything, right?
Yeah, because
Yeah.
you're telling us that like when we're like running, we
Yes,
use a lot
that's
of
right.
oxygen, but then we start running out of reserves.
We go into anaerobic mode and we have lactic acid. That's
Yeah.
during the Krebs cycle, exactly.
We
Right.
make a bioweapon in class.
We will not be making a bioweapon in class.
Darn.
Yeah.
Darn.
Okay, well, it shows here, right here, in the starch in our stomach, it goes in here, it's making certain things. And in fact, there's a time during the fermentation process, if you remember, one of the things that's a byproduct of fermentation is gas. And so if there's certain foods that you eat that make you, quote unquote, gassy, like beans or whatever, it's because you've gone into the fermentative process. It's not a problem. It's just your body has to deal with it some way. And one of the ways they deal is to release the gas.
Could you use bean gas to cook stuff with if you like lit it on fire, you think? If you like let it sit and like ferment, you could like keep it
Just collect
trapped
it,
somewhere. You could put it in like a propane thing that's empty.
Technically, it will burn.
You
Yeah.
could like burn it, like okay.
Technically, yeah. All right, so conditions for
Remember that
bacteria
pretty
growth.
complex.
Okay, so bacteria, remember the number one problem with bacteria is desiccation or drying out. So how do they grow well? Here's the question, right? They need to have the proper amount of moisture. So moisture's number one. I think you should have a chart here that you're filling out things that bacteria need for optimal growth conditions. Okay, moisture's number one. Moderate temperature, whatever it is, not too cold, not too hot. Now, basically, we're talking about bacteria here, the regular old everyday bacteria, not those extremophiles. Remember the extremophiles one, the archaebacteria can live in bizarre places like the caldera of a volcano where it's very hot, or in
Isn't it-
ice where it's very cold.
Yeah, they found stuff
Absolutely.
like really deep in the ice. still
Exactly.
functioning.
So this is more talking about the, let's say, everyday bacteria here, okay? We've got adequate nutrition. They have to have something to feed on, whether they're photosynthesizing, chemosynthesizing, or going through heterotrophic respiration through the process of eating or however they're going to absorb their food. They prefer darkness. They like it to be dark, not too much light. It turns out the light of day, UV light, does kill bacteria. It's true. You probably heard something about that during the COVID time,
Yeah.
right? Now probably sunshine because of vitamin D conversion in your skin. Vitamin D is so good for our bodies to keep us from getting sick. But literally, when they talked about shining lights into your lungs and stuff, and people made fun of certain people who said so, it's true. You put lights down, you will kill bacteria.
Wait, isn't COVID not like a bacteria?
It's a virus.
It's a virus. Can't you not kill a virus?
Because it's
though?
not alive.
It's not
technically.
alive,
You
which I
can't-
was like.
Yeah.
kill
We'll talk
COVID
about
though,
viruses
but, like,
today, and I had like whole courses on virology. It's very interesting, but they're not considered alive, and we'll talk about it. It's because they don't have a form of reproduction. They have to use a host cell for reproduction. Yeah.
So bacteria is like Batman.
Wow. Okay.
Well, Batman likes being in Gotham when it's raining. moisture.
Okay.
He likes us when it's a little bit cold, but not too hot, a moderate temperature.
All right.
He has a very balanced nutritional diet.
He does? Have you seen him eating?
Oh yes,
Really?
many,
He
many
said
times.
that very creepily. Oh yes, I have.
Uh-huh.
He's a stalker.
And he,
What's the last one? Proper amount of oxygen.
I think
I
that
think
works.
he
for
works
that.
out.
Yeah.
Okay, that works too. Okay,
And he
Batman.
likes the darkness.
So notice I didn't just put oxygen because we know there are some anaerobes out there, right? That need to not be in oxygen. I'm going to say the majority of bacteria are aerobic. They need oxygen. But there are those weirdos that really grow in anaerobic environments. And they, like I said before, tend to be some of the
Bad ones.
really, really pathogenic ones. Yeah. All right, 1, 2, 3, 4. We have 5 things. You'll have 5 things written down?
Yep.
And that's all the blanks there? Yeah? Because then the next one is,
There's another.
how do we keep them from growing? Because if we want to keep bacteria, say, out of our food, for instance, we want to limit the amount of growth that's taking place there, okay? So number 1, we'll cook it. Cooking almost always will take away pathogenicity from bacteria because cooking will denature it and kill it in most cases. There are certain cases where the bacteria will be killed, but there will be a toxin left over. The toxin does not get taken out by cooking. All right, and there are certain ones I'll talk to you about in a minute about that.
So people like in especially in third world countries where they have to boil the water or they'll die. Does that mean it doesn't get rid of the bacteria?
The boiling does get rid of it. That's why they boil it.
Yeah.
That's why they boil the water. Yeah.
That's why we
And
cook.
somebody just recently in nearby because of water pipe breaking, I think Hays County, right? They were all told to boil their water because they couldn't guarantee that it was treated properly because there were some pipes broken. And I know during the big freeze 2 years ago, we had that in Bastrop. We had to boil
just
water.
we had 3 pipes break
Yeah.
just down the road from our house.
Yeah, there were big ones in the whole city. Yeah, and we had to boil.
That's why
Chloe,
we,
you have a question? No. Yes.
That's why we cook raw meat and like more fresh stuff.
Most people cook meat. Yeah.
Yeah.
So some people
Depends
do it
on
raw.
if it's steak or not.
Yeah.
Very specific.
I like
That's
it.
even though
Agree.
even if we like
Right.
freeze them, like kill an animal, freeze the meat. So
Yeah, that's
cook it.
another way of doing. Okay, canning is another one. So think about it. Have you ever done canning?
Yeah.
Mhm.
The idea here is that we don't usually can. We usually use jars, right? You know. But the idea here is the goal when you do this like this is to get all the oxygen out of the jar, right? You clean the jar really well to start with. It's a beautiful picture actually for like salvation. Your jar gets clean, then you get filled, then you get sealed. So filled with the Holy Spirit, sealed. You know, and we're all clean on the inside. You know, that's kind of a cool deal right there. So canning.
Pickles.
Like pickles. We're like pickles. Jesus is like a jar of pickles. There you go. Dehydration. So beef jerky, right? We take all the liquid out. Dried fruit, things like this. The stuff the guys took to space, right? They took dehydrated food. Because if there's no water, guess what? Bacteria dry out. Enemy, they can't grow. Now, could there be some spores in there that could be reactivated by moisture? Sure. But if it's food, we'd probably eat it and our body would take care of it. Our acid in our stomach would take care of it. Freezing. So you brought that up, Eddie, about doing that, putting it in the freezer. That'll do it. That'll stop everything. So a lot of times at Thanksgiving time, people say about your turkey to be sure that it's cooked properly. And everybody's always worried about the turkeys. I've never ever had a turkey make me sick. And I let it thaw like in the fridge. I put it in the sink overnight.
That's what my mom does.
It does fine. Yeah. So, but then we can take it and freeze it. So let's say we have some meat that's starting to go bad. It's just starting to just get old or whatever. There could be some growth in there. We can put that into the freezer and that'll take care of that. But if there are spores and you pull it back out and let it thaw, it might have some regrowth in that. All right, so put fresh stuff in there. There's a process called pasteurization. It's named after Louis Pasteur, who was one of my heroes. He's a microbiologist. Did we talk about him in the last time? He's the guy that saved the wine industry in France, yeah? He's also did a lot of work with viruses in the sense that he worked with rabies, kids who had children who got rabies. All right, so it's heating something up to 161 degrees for 15 seconds and cooling very quickly. Yeah, cooled immediately. We do this almost with all of our milk, unless you're drinking raw milk. You can't buy raw milk at the grocery store because of this problem. If you're drinking raw milk, it's really good for you. I have no problem with raw. It's great. You just need to know where it came from and when it came out of the cow. Okay, so just keep track of that kind of thing.
My grandma did goats.
Goat milk,
Fresh
yeah.
goat milk is really good.
Is it?
Eat
Okay.
that chunk of milk.
My grandkids all drink raw cow's milk that they
We
get from
used to
some
do that.
farm over here somewhere.
We
huh?
used
Yeah,
to
there's a
do
bunch of
that.
cattle farms
We
Yeah.
have
towards
a
Smithville.
we have a deal with these people that we drink the milk and we pay them.
Uh-huh.
And we did that for like 3 months, and like it was really fresh milk, and it was like really good. Except it was so chunky. Like there were
Yeah.
so many chunks in there.
I'm gonna say I'm not fond of milk with chunks in it. That doesn't.
Yeah, we
sound
switched back
appealing.
because we're like,
don't like
we're
milk.
like, this is good.
Well,
milk,
that's
but it's
probably you
just
could have made
so-
cream out of it.
I think we did.
Okay, yeah, because it floats to the
These
top
are
usually.
5-gallon
I know
buckets
my
of whipped
grandkids, they just take
cream all
it,
over the
shake it
house.
really well, and they
Yeah.
love it. It's wholemeal.
Lots of ice cream.
I just
Ice
drank.
cream.
Strain it. That would
All right, asexual reproduction in bacteria. This is essentially how reproduction takes place asexually. They make a copy of themselves using a process called binary fission. You have one bacteria, it duplicates everything, and you have two bacteria. It's very efficient that way. It's actually a very efficient way to have reproduction. So having sexual reproduction is actually a step back away from this, making things less efficient. And so the simplest life out there has the most efficient reproductive process.
So we should all clone ourselves.
This is cloning is a good word for it.
Yeah.
It is a clone.
Interesting.
It's simply a clone. Now we have it in our bodies, of course, like our skin cells, they're clones. They just keep, going through mitosis. All right, a DNA loop attaches to the plasma membrane, makes an exact copy, just like when we go through cellular reproduction in regular cells. The DNA gets duplicated. The cell wall elongates, separating the two strands of the DNA. And then a new cell wall material membrane grows, is made, and an exact copy is made. There's that word right there, clone. It can happen in only 20 minutes. Bacteria can grow really, really fast. And because of that, and if they're infecting, like say your throat with strep throat or something, boom, it can all of a sudden just take off and make you really sick really quick. So there's something going around right now, right? So maybe it's bacterial, probably viral. Virus is a little different. Let's watch a little video on this reproductive technique.
Asexual reproduction in bacteria starts when the DNA of the bacterium forms a loop and attaches to the plasma membrane. The DNA loop is then copied. This makes sense because we're going to make another bacterium. To make another bacterium, we have to have another set of DNA. So now the bacterium has two loops of DNA. To separate them, the cell wall begins to elongate. That pulls one loop one way and the other loop the other way. As the two loops get farther apart, the cell wall and plasma membrane begin to constrict. This forms two cells where there was originally one. In the end, then, there are now two bacteria, and they are both genetically identical to the original bacterium that started the process.
All right, quite simple. Exponential growth. So this is how, when we're going through this regular reproduction right here, we have one cell becomes 2, becomes 4, becomes 8, becomes 16. This is all happening 20 minutes, and boom, you have this massive exponential growth of a colony of bacteria. Yes.
I'm glad this isn't how reproducing works for human. I don't think I'd want 16 if you
No,
that
we
would
just
be
cloning and
terrible.
boom, they just
That'd
fall
be
off
horrible.
of us, Oh, yeah.
That would be horrible.
So perhaps, sexual reproduction is a way to curtail the population. It's unhindered because of the abundance of resources. As long as there's enough resource, enough food source, and that we have the right amount of oxygen, we have the right temperature, that kind of, as long as this is happening, we can have unrestricted growth. All right, now, because they're all clones, that presents a little bit of a problem. That problem is a lack of diversity. All right, so we don't have any genetic diversity here. So if there's a particular trait that something exhibits, it won't have any chance of growing past that. It'll keep producing that particular trait, whatever it happens to be. And so there is a way to get some DNA diversity into bacteria. They have a couple of strategies to do that. So they can exchange helpful genetic information in 3 different ways. We're going to be looking at conjugation. We're going to cover these in some details in a minute. We have transformation and transduction. Okay, so 3 processes here: conjugation, transformation, transduction, and we're going to go through each one of these individually so you understand what's happening here. But this looks like a good quiz question. What are the 3 ways that bacteria can achieve diversity genetically?
CTT.
CTT, right there. All right. Does everybody have those 3 words written down? Maybe put a star by them. And they may be just listed separately in the book, right? So when we talk about them separately, I would know them, is my point. If I were you. We'll have a quiz next week, right? That sounds right. Okay. All right, so here's transformation. We're going to see what happens here is that a transfer of DNA segment from a non-functional donor cell, essentially a cell that's dying, will send out a little chunk of DNA to a donor cell. This is the donor cell, to a recipient cell. Look, it'll integrate itself into its DNA. Okay, the living bacterium will absorb the trait it needs from this dissolving bacteria. Okay, interesting. So that's transformation. Adding that in, you would wonder if this might be a evolutionary tactic, right? When they talk about evolution, adding in a new gene. That's interesting. So my question has always been, how does the recipient know it needs that part? Why does it take that
Probably
one
one
little
thing
part in
that
there? Why
it
that one
doesn't
and not
have.
another one? So I find that interesting.
I think this has a very scientific answer.
Telepathy.
Exactly.
Telepathic, okay. Telepathic transformation. Transduction, the process in which, this is interesting, a virus comes in. This is a special kind of a virus. The virus is called the bacteriophage or bacteriophage. And it looks like a little spaceship. It actually injects some DNA because remember, viruses, we'll talk about them. Viruses have DNA in them, maybe RNA, depending on what the virus is. I think COVID was an RNA virus. But they need to reproduce inside of a cell. So here we see that the virus actually infects the cell. The DNA is transferred from one bacterium to another, right here. And then the plasmid, a small circular section of extra DNA, confers a unique trait separate from the main genetic code. So we have a separate little deal here. I thought I had a video on this one. I might.
It looks like spiders.
It does.
They do look like little
it looks
spiders.
like that little creature from like the first or second Star Wars that latches on.
It looks like a little
spaceship.
hypodermic needle, honestly, because it's got a protein coat up here and then this long neck
It looks
with
like
a
on
little
the
injection
Gen
area.
Q robot things.
Oh yeah, yeah.
So it's like
Cookie bots.
the microorganism's version of a mosquito.
Okay.
Yeah.
I like it,
there you
but it
go.
goes through 2 different cycles, the lytic cycle and the lysogenic cycle. A lytic cycle means lysis will take place. Lysis means it's going to burst the cell, it's going to kill the cell. So in this case, the virus kills the cell. But it does confer that little bit of genetic information in there, which will be passed on. Possibly it could be beneficial, sometimes not. Let's watch a little video on it.
Bacteria engage in horizontal or lateral gene transfer, meaning that genes are exchanged between cells of the same generation. In contrast, vertical gene transfer occurs when parents pass genes to their progeny. The 3 methods of horizontal gene transfer that bacteria employ are conjugation, transformation, and transduction, with conjugation being the most common. Transformation and transduction typically take place between bacteria of the same or closely related species. Transformation occurs when bacteria take up extracellular DNA and incorporate it into their genomes. Typically, this occurs when one bacteria lyses, or splits open, releasing its genetic contents, and then another bacteria comes by and acquires it. Bacteria able to bind large amounts of DNA are termed competent. Competency is a state of increased cell wall and cell membrane permeability that allows cells to uptake DNA. Many bacteria are naturally competent, and so actively bind environmental DNA. After transport into their cytoplasm, the bacterium can incorporate the new DNA into its genome through the process of recombination. Recombination is the rearrangement of donor and recipient genomes into new hybrid genomes. This can result in new phenotypes. For example, the bacteria can acquire pathogenicity or antibiotic resistance. Some competent bacteria actually kill non-competent bacteria to release DNA for transformation.
Oh, steal.
Transduction occurs when DNA is transferred from one cell to another by a bacteriophage, a type of virus that infects bacteria. Viruses cannot replicate on their own. They are obligate parasites that rely on host machinery. Many bacteriophages can switch between a state of lysogeny and a lytic cycle. When the bacteriophage is in a state of lysogeny, the virus combines its genome with the bacterial chromosome. The viral genome hangs out there for many generations as the bacteria replicates. When induction occurs, the virus switches to the lytic cycle. The cell becomes a virus-producing factory until it gets so full of virus that it lyses, or bursts open, releasing virus particles into its surroundings. There are two kinds of transduction, generalized and specialized. During generalized transduction, the phage capsid accidentally assembles around a fragment of bacterial DNA or a plasmid. When the assembled viral particle infects a new bacterium, it injects its previous host's DNA into its new host. Specialized transduction occurs during the lysogenic life cycle of the virus. Incorrect excision of DNA during induction results in a fragment of bacterial DNA being picked up instead of a part of the viral genome, which then stays in the bacterial nucleoid. Now, the bacterial DNA replicates as part of the bacteriophage genome, is packaged into phage capsids, and is injected into new bacteria. Horizontal gene transfer, along with mutations, allows bacteria to achieve genetic diversity. These processes allow bacteria to adapt to new environments because they enable them to respond to selective pressures. Mutation is a slow process, and most mutations are harmful or neutral, not beneficial to the bacterium. Meanwhile, horizontal gene transfer is a rapid way to acquire large chunks of DNA from another bacterium all at once. So if bacteria keep acquiring new snippets of DNA from other bacteria, do their genomes keep getting larger and larger? No. If the transferred genes don't provide a selective advantage, they are usually lost by deletion, so the genome size remains constant over time.
All right, so over time, then, it's constant. It's the same one. All right, conjugation. This is the most common. Isn't that what the video said? Conjugation was the most common? Yeah, this is the most common form right here. The temporary union of two organisms for the purpose of DNA transfer. So, for instance, if one of them has a plasmid, it can actually copy that and send that information through this called the pilus or the pili. Remember we talked about pili? Those little kind of finger-like structures on the outside that make it sticky sometimes. So sometimes they're called sex pili because they're gonna confer some information. It's not true sexual reproduction, however, because the whole genome did not replicate. Okay, it's just sending a little bit in there. So it'll take that, but it will copy it. It'll open up this little temporary doorway. It'll send it in, and the other one will have it there. So a plasmid is a small circular section of extra DNA that confers one or more traits to a bacterium. It could have been acquired, by one of those other processes, including viral infection. It's initiated by the donor. The donor comes up and says, hey, I got something you might like, and it sends it that way. And so I have a video to kind of sort of, you didn't have to write a bunch of stuff down on this one.
If a bacterium has a desirable trait on a small circular strand of DNA called a plasmid, it can transfer that trait to other bacteria by conjugation. This process starts when the bacterium that has the plasmid approaches a bacterium that doesn't have the plasmid. The two bacteria then grasp each other with their sex pili. A conjugation tube forms between the bacteria, and one strand of the plasmid transfers from the bacterium that has the plasmid to the bacterium that doesn't. Because of the properties of DNA that you will learn about in later modules, the bacteria each can reproduce the second strand of the plasmid by having only one strand each. When the plasmid is fully transferred, the conjugation tube collapses, and the result is two bacteria, each of which now have the trait conferred by the plasmid.
And that gives diversity now. So we can take that diversity from one to the next one, and it confers that over to this next one over there. In the video where he said you'll learn about the process in a future, well, that was back in the old book. We've already learned that process. That's the whole replication of DNA situation. All right, in your book you will probably be reading somewhat about this thing called biosphere out in the desert somewhere, I think in Arizona. Could be New Mexico. I think it's Arizona though. They wanted to build this facility that would be completely and utterly self-sufficient, where they could have all these plants producing oxygen, and they could grow everything they needed, and they wouldn't need any anything from the outside world to be put into it. So they came through here, but they did not calculate properly the amount of bacteria they needed. Because it turns out, remember where the vast majority of our oxygen comes from? It comes from the algae in the ocean, blue-green algae, which is bacteria, and other bacteria going through their processes. So it was an absolute failure, but I will say that it was interesting. Okay, it wasn't a failure. We learned a lot from it. Really, science is about learning something, so there's really no failing. I will say in this particular case, there was a little bit of arrogance involved. We can make our own self-sustain. No, they couldn't, because they couldn't replicate what we have here on the earth, right? You know, they couldn't do that.
They start speaking different languages after.
Their Tower of Babel situation going on there. Furthermore, there are human beings in there, right? And human beings have different, I don't know, motivations for things. Let's watch a little video about it.
This is Biosphere 2, a supersized 3-acre terrarium. Originally funded by Texas billionaire Edward P. Bass, its 6,600 panes of glass enclose 7.2 million cubic feet of space, including an artificial rainforest, desert, and tiny ocean, in addition to 3,800 species of animals and plants. When it opened in the early 1990s, it was hailed as one of the world's most impressive scientific undertakings. But it quickly became one of the most notorious, too, as the home of the Biospherians, a team of researchers who were sealed inside for years at a time in this supposedly self-sustaining environment and left to fend for themselves. Their mission was to use their world to develop new technologies that might help humans survive on the Moon and Mars. They vowed they would grow all the food they needed, recycle their water, air, and waste, and live without supplies from outside. But reports soon surfaced that the Biospherians weren't staying true to their word. Stories that they were actually surviving on pre-packaged food they had hidden inside, secretly replenishing their oxygen from outside, and using CO2 scrubbers to prevent the buildup of dangerous gases. The reports set off a scientific scandal. Though it was rumors about what members of the mixed-gender team were doing after work that really caught the world's imagination. Rumors seemingly verified when 2 of the team members got married almost as soon as they got out. What happened in between might have been perfect for reality TV. Unfortunately, the gossip and scandal ended up overshadowing the serious research being done here, and even helped bring it to an end. Today, Biosphere 2 is still trying to live down its soap opera past and live up to its founders' scientific intentions.
Okay, so what's the one reality show, Big Brother, where everybody's locked into a house? It's a Big Brother? Does that sound right? Where everybody's locked into a house and then they, you know, somebody gets I don't watch any reality TV, so I don't know what they are.
I don't know what it is.
But there was something similar to that, but not trying to survive. So literally, they had to like open windows to get air, and there was not enough oxygen. And
Survivor. There's a show called Survivor.
Yeah, but they're outside. This one, they're all like locked in their house.
Oh.
It's a nice house with a pool and that kind of stuff.
I've watched Phineas and Ferb.
I don't know.
There you go, reality TV.
All right, here's what I want to tell you though. The facility is still there, and they're using it now for research, and they have summer programs that students go to. I had a student of mine come to me and say, hey, look, I want to go to a week like camp or whatever at the Biosphere Project, but it's fairly high level, you know, snazzy stuff. They needed to have a teacher recommendation. So he needed my recommendation. I wrote a letter saying he'd be great for this, and he went and spent a week there, and they did various experiments and stuff like that. So that's pretty cool if you want to look into it. Yeah.
Technically, they didn't completely fail. They created in life.
Well, it said they got married. Didn't say anything else. Yeah.
Okay.
All right. So it says here, remember that most bacteria are beneficial. Hooray! So they get kind of a bad rap for the superstars. The superstars are the pathogenic ones, right? But don't let that fool you. Most bacteria are beneficial.
I don't trust it.
Bacteria are chemical recyclers. They can be used in something called microbial bioremediation. When I was in college, one of my instructors, one of the things he was developing was bacteria that would eat up globs of oil, for oil spills in the Gulf, and this kind of thing. Very strange. They said it had nothing to do with his research, but he ended up being rendered a paraplegic. He was in a wheelchair. I remember, so I had him as an instructor in my last class I ever took with him. He was in a wheelchair, and he said it had nothing to do with his research, but I find that a little bit suspicious that some weird thing happened that made him a paraplegic. I don't know, but he did help develop this whole idea of the bacteria eating up oil spills. The gut. It turns out good bacteria in our gut synthesize vitamins K and B12 in addition to helping digest our food. I am so happy that people have finally figured out the microbiome of our body is super important, because everybody thought bacteria, all these years since I've got my degree, I'm always like, bacteria is our friend. And people are like, but it causes disease. You know what? Honestly, there are two big competing ideas between pathogenicity caused by the germ theory versus the terroir, meaning your body. And I think COVID really, really turned the spotlight on this, and that's the idea. If we have healthy bodies, we can avoid bad diseases, right? Our vitamin D. We all found out during COVID times that everybody was deficient of vitamin D, right? We're not getting enough sunshine. And I don't know about y'all, but I take it in a pill now, right? All the time, especially in the winter. Yes?
Do you know where we're supposed to write that in the book?
I have it underlined, but there's no place for it. If there's no place for it, then don't worry about it. This might be me just preaching to you about the importance of the microbiome. All right, so let's watch this TED ad about it. I love it. I love it.
Being human. We each view ourselves as a unique and independent individual. But we're never alone. Millions of microscopic beings inhabit our bodies, and no two bodies are the same. Each is a different habitat for microbial communities, from the arid deserts of our skin to the villages on our lips and the cities in our mouths. Even every tooth is its own distinctive neighborhood. And our guts are teeming metropolises of interacting microbes. And in these bustling streets of our guts, we see a constant influx of food, and every microbe has a job to do. Here's a cellulitic bacteria, for example. Their one job is to break down cellulose, a common compound in vegetables, into sugars. Those simple sugars then move along to the respirators, another set of microbes that snatch up these simple sugars and burn them as fuel. As food travels through our digestive tract, it reaches the fermenters who extract energy from these sugars by converting them into chemicals like alcohol and hydrogen gas, which they spew out as waste products. Deeper in the depths of our gut city, the syntrophs eke out a living off the fermenter's trash. At each step of this process, energy is released, and that energy is absorbed by the cells of the digestive tract. This city we just saw is different in everyone. Every person has a unique and diverse community of gut microbes that can process food in different ways. One person's gut microbes may be capable of releasing only a fraction of the calories that another person's gut microbes can extract. So what determines the membership of our gut microbial community? Well, things like our genetic makeup and the microbes we encounter throughout our lives can contribute to our microbial ecosystems. The food we eat also influences which microbes live in our gut. For example, food made of complex molecules like an apple requires a lot of different microbial workers to break it down. But if a food is made of simple molecules like a lollipop, some of these workers are put out of a job. Those workers leave the city, never to return. What doesn't function well are gut microbial communities with only a few different types of workers. For example, humans who suffer from diseases like diabetes or chronic gut inflammation typically have less microbial variety in their guts. We don't fully understand the best way to manage our individual microbial societies, but it is likely that lifestyle changes, such as eating a varied diet of complex plant-based foods, can help revitalize our microbial ecosystems in our gut and across the entire landscape of our body. So we are really not alone in our body. Our bodies are homes to millions of different microbes, and we need them just as much as they need us. As we learn more about how our microbes interact with each other and with our bodies, we will reveal how we can nurture this complex invisible world that shapes our personal identity, our health, and our well-being.
So how many are crawling on my face right now?
Millions. And you should be grateful for them. It's keeping you from having a fungal infection on your face. Yes.
So I mean, there's tiny cars just zooming around our
You want to
sun.
think of them like cars? You can. You can. All right, so the idea here is the idea of probiotics. All right, it's become very popular recently. It's the idea of replenishing your gut bacteria by taking a pill. I take a probiotic every single day, and I'm taking millions of bacteria into my body. Furthermore, if you eat yogurt, y'all like yogurt?
Yes.
No, I'm not a fan. Not a fan. Fermented foods are really, really good for us. We've come to find out. I'm just not a big fan of fermented foods.
So,
Kimchi's the one I can do, and
I
it's
like
me. I don't like, what's the kind that you make
I guess
that
it's
The
you,
kombucha.
oh, I can't
Kombucha?
stand
I don't like
kombucha.
Oh, my mom used to make all sorts
Ooh.
of like gallons of it. It's
And that
good.
giant thing is floating in the
Oh, yeah,
mother,
it's good.
you know.
So
It looks like a
like-
I don't like
jellyfish.
yogurt either.
I
I think
can-
it smells bad.
I don't like the,
I like yogurt.
the tartness or
I
whatever
can't
that is.
stand
Well, I like
It's-
So like wine?
Like the sweet.
So
Like it's
wine
from
is
fermented
fermented.
berries.
It's a fermented food,
Maybe
but
like
not.
without alcohol.
Well, it has alcohol. Yeah, it's part of the fermentation process. Yeah. Typically, there's no bubbles left though. They let that go out of the wine, unless of course it's champagne. They keep the bubbles in.
Does ale have the bubbles in it?
Say again.
Ale.
Yeah. Yeah. They let the bubbles out. You know. So I don't know if they have any sparkling beer product. I don't know about it if they do.
There's such a thing called biotics, the pros and the cons.
No.
Oh,
Except
okay.
eating. How about eating junk food or eating like highly processed food? Highly processed food doesn't challenge your gut to keep those healthy bacteria growing. So you really need to challenge your gut by things that are quote unquote hard to digest, like complex vegetables, stuff like that.
Are dried fruits and like, do they go through the fermentation process or is that a different process?
Dried fruit, the drying is to keep it from going through the fermentation process. Yeah, it's to keep the oxygen out of it. You know, it's gonna keep it from doing that. So it's a preservation method. Okay, how do they get insulin? Okay, so they can actually use bacteria to produce insulin for diabetics who need to have insulin injections every day. So used to they would get it from some sort of animal or something, and now we can actually use bacteria to generate it that way using something called recombinant bacterium to go through the fermentation process and to release a bunch of insulin. Then they can take that insulin and put it in a vial, and people can inject it in themselves. These are people who have diabetes. Okay, so we can see how does insulin work? The idea of insulin is that it opens the door to let the glucose inside your cell. So it helps to escort glucose into your cell. Do our cells need glucose? Think about crabs. Crab cycle. We need that glucose, right? So it's got to get in. So people who have diabetes either have a lack of insulin to open the door, so use the key to open the door, or they have a lack of doors to be open. And it's the second one is called diabetes type 2, which is more common. That's those people, if you probably know somebody who has diabetes type 2, maybe a grandparent or something, because as we get older, sometimes our doors stop working, if you will. And so what they have to do is eat kind of not large meals, but smaller, like grazing meals over the day, so they can keep everything all evened out and not overload
My
the little
brother has
receptor.
diabetes. I'm not sure whether it's 1 or 2, but I know he has.
You're who?
My brother, Daniel, adopted brother.
Okay, I was about to say you don't have a brother.
Adopted, yeah.
Okay, I know Daniel. Yeah. Okay. Food production. Here we are in yogurt. Now, we talked about yogurt a second ago. It's very good for you, but it has to be a live culture yogurt. There are some yogurts out there that are just full of sugar and they don't have a live culture in them. And I think the ones they give the kids that you squirt up out of a little tube.
Tubes.
I mean, come on, that's just full of sugar.
What about
So Greek yogurt is the one you're supposed to eat. Yeah, and unflavored, unsweetened Greek yogurt.
So none of the other ones would really
I
have
like to
it.
put some jam.
If you got to put something in it, some
My-
fruit
my
or
mom puts
whatever,
coconut powder in
to do
hers.
something. Some people use it
Pretty
as a
good.
substitute for sour cream. I could see that.
Oh yeah.
on tacos?
What do you
Well,
think?
It would
with
have to be
Greek
a
yogurt,
very
if you
Greek
add
yogurt.
the ranch powder, you can make it in a ranch, and it's a lot better than
Oh,
like
is it?
store-bought ranch. It is
Okay.
so much better.
Just using Greek yogurt instead of milk.
Yeah, and you just put ranch powder, and like you get the package from the store in the Greek yogurt, and it's so much
That
better
sounds good.
than
Yeah,
it
as a
really
dip
is.
or as a salad dressing?
You can use. I've used it as both. I
Okay.
prefer it as a dip though, 'cause it's thicker.
It's
than-
thicker,
So
right.
we would be the most
Ooh,
healthy
cheese.
humans on earth if we just only ate Greek yogurt.
No.
Well, you need some
Eat
more,
honey.
but in addition to some things.
Protein.
But yeah, but eating fermented foods is a really good idea. Supposedly cheese is like the perfect food.
Really?
'Cause
That's
it's
what they
protein
say.
and
Bad.
I love cheese, but I feel like it makes me fat. So I don't know. I think it's too fattening.
It also
I
gives
feel like
you
it stops some of my
constipation.
systems.
Oh, okay.
It's not like
Stops you up.
Just more.
Yeah. So here we can see different kinds. Right here,
Yeah,
we're going to
blue
talk
cheese.
about Roquefort TI soon. I think. Yeah.
Maybe a little off topic, but how can we eat cheese that is moldy?
I know. Isn't that interesting? People do. It's called blue cheese.
it's not.
The
good at all.
blue
It tastes
is.
disgusting.
We're going to talk about it next module because I believe that it's a fungus growing in there. Yeah. So we
Give
find
it
it.
to me.
Technically,
and
all
something
cheese has a lot of bacteria in it.
That's the process by which it's made.
Yeah.
Absolutely, 100%. Here, what is that?
It looks like
Whipped
mayonnaise.
Oh, maybe that's sour cream.
Sour cream.
I
Oh,
thought it
that
was
would make
ice
sense.
cream.
Yeah.
It
Yogurt,
looks
cheese,
like
sour cream. I like sour cream a
I
lot.
thought that was ice cream. I was like, I'd love to eat ice cream every day.
would take
It's
Greek
not
yogurt.
fermented, sorry.
Oh.
Okay, here's the fermented foods that we should be eating more of. Okay, ready? Kimchi. That's the one I can stand.
What is
I'd
Wait,
be okay with
I like
that.
pickles.
And
I could eat pickles.
fermented cabbage.
Yes.
Interesting.
Cabbage. It's kind of got a little heat to
If they
it.
put spices in it.
Yeah. Kombucha, tempeh, I don't know that one. Kefir. It's kind of like
Ooh,
liquid yogurt.
Yeah.
Yeah,
It's a little
sauerkraut. Now I can do sauerkraut, and it's got to be the fermented type, though, not the kind in the can that's been heated so much that everything was killed. So, but I think HEB sells an organic fermented sauerkraut. Yogurt, that's live culture. Do y'all have a place to write this, or are you
I
just
don't think
finding a
so.
spot? Okay, okay. I didn't think. I thought I was just preaching at this point instead of making you write notes down. But pickled vegetables, I do like those.
I love pickled vegetables.
Pickles are good.
Pickles
Pickled
are
okra.
tasty. Oh, pickled okra is amazing. With like
Oh.
jalapenos.
Oh,
Not the
to
biggest.
try that.
I'm not the biggest fan of pickles.
Oh, do you
I've
like
had
vinegar?
A lot of times when I get cramps during games and stuff,
Pickle
Oh.
I
juice?
have to drink
Make you
pickle
drink
juice. I'm like,
Yeah,
"Ah."
mine was the same way. All right, enzyme production. Okay, so we can have it. We can use bacteria to produce certain enzymes that we need for industrial applications. We just make it go through the process of the metabolism of the bacteria, and we can end up with something desirable from that. So now, here's what I'm trying to enforce that bacteria can be good. Did I talk about the pathogenic ones during the video last time?
I think you touched on it a little
Or am I
bit,
way
but-
behind? Medicine! Looky there. We have bacteria that are used to make some certain medicines. Oh, here we go. Let's talk about the pathogenic ones, the bad ones, okay? So, probably the most virulent bacterial infections that happen come from this group of bacteria called the Clostridia. All right, so this one right here we're looking at is called Clostridium botulinum. And in the old book, y'all had to learn this, Firmicutes firmibacteria. Firmicutes is its name of its genus species. But I don't think this book requires you to do that anymore. Yes,
Is that the botulism one?
it is.
Okay.
All right, so here's what happens. This, by the way, is an obligate anaerobe. What does that mean?
It does not need air.
It's more than that. It cannot grow in air.
Cannot grow in air.
Air kills
Air
it.
repellent.
Yeah, air kills it. So it has to grow in an anaerobic environment, which is good news, right? Because as long as I'm not in a place where there's no oxygen, I'll be fine with this. Here's the problem. What happens with Clostridium botulinum is during the process of metabolism, it manufactures and releases a toxin called botulinum toxin. And that botulinum toxin is the deadliest thing we know of a biological nature. Okay, it's not the bacteria that kills you, it's the toxin that it releases. So if this bacteria, say, is growing in a jar of green beans, all right, the jar of green beans has no oxygen in there, right? That's because we canned it and we got all the oxygen out of it. Well, if, say, some of this bacteria came from dirt or something, that's typically where we find it in dirt, way down deep somewhere. It's kind of weird how it would get there, I don't know. But here's the deal. As it's growing, it makes this toxin. Let's say I open the jar and I cook it. Well, the cooking kills the bacteria. The oxygen kills the bacteria, but it doesn't kill the toxin. So the toxin's still there. And that toxin, they say the amount of toxin that fits on the head of a pin, like a sewing pin, you know, a little tiny top of that, 10,000 men can die from that. It's very, very powerful poison.
So
All right.
never eat canned foods again.
Well, so have you heard the idea of a can that gets bloated, like popped out like this? So part of the process of metabolism also produces a gas.
So if you see a bloated
If it's a bloated
don't
can,
eat it.
stay far away. Yeah, stay far away from it. Because that could be the case, that there is botulism in there. You don't want to eat that, okay? Even if you cook it. Not a dented can, a bloated can. Yeah.
So to someone you don't like, you could
Oh, this would be
like,
terrible.
this would be, I made this jar of pickles for you.
Not only is it deadly, it's a painful way to die. Well, okay, I'm gonna take it back. It's not as painful as as another one in Clostridium tetani, which is tetanus. Tetanus is terrible.
I've got it.
What happens with botulinum is it causes every muscle in your body to relax. So you might, oh, that's not so bad. Everything relaxes, including your heart muscle, and you die.
Can
So-
you survive by CPR?
It's a toxin that's hard to get rid of. But here's what I'm gonna tell you. We use it medicinally.
Really?
People use the toxin of botulism, botulinum toxin. They inject it into people's faces to make the wrinkles
Botox.
go away. It's Botox.
Really?
So think about it.
I thought it was
If I'm
just
going to relax
plastic.
everything, I can put it in my little wrinkle. It will relax my wrinkle, and my wrinkle will go away. But of course, everything will be paralyzed right then.
So
while
wait,
the toxin's still
if
there.
you got
So you can't
Botox
feel your
enough, it
face
would
after
kill
that.
you?
Yes. I couldn't believe it when people said, "Oh yeah, we're gonna put Botox and inject," but what? You're injecting deadly toxin into the
So
body.
you not feel your face after that?
Yeah. Like if you
That
ever see
area?
somebody who their forehead doesn't move, you know, and they probably had Botox in there to keep their little
That's-
forehead,
ugh.
you know, like
So
that.
Botox is different than plastic
It's a
surgery.
very dilute form of the toxin, but it is the toxin. It is it. Oh, they've been doing some research on that toxin with migraine headaches.
Oh, really?
And having some success with it. Yeah, but they're using that very deadly toxin to do it. But it's going to relax whatever's causing that pain to take place. Now this is in the same group. Look, Clostridium tetani. Okay, so this is tetanus. And you don't want to, in fact, I have often said that if you're going to get one vaccination in your life, like you're anti-vax or whatever, I'm cool with that, don't worry. But if you're going to get one, I would get a tetanus vaccine because you do not want to die of tetanus. It's bad.
I've had tetanus shots. I've had like 6 or
But what did
7
you do? You stepped on a
I've
rusty nail.
stepped on metal. I've had it way too many times.
Okay. It is an
Oh.
obligate anaerobe. It can only grow where there's no oxygen. So typically where you find it is underground, again, deep down. I've always thought it was weird they say you step on a rusty nail.
Yeah.
Well, for a nail to get rusty, it had to be exposed to oxygen. So I don't quite get the rusty nail part of it. But if you're digging around deep, you know, and you end up getting punctured by something, yeah, that's how you can get it.
Like working with pipes or something
Mhm,
underground. If
mhm.
you're working on that, you get cut.
Digging
by a pipe.
trenches in a war, for instance. Yeah.
I stepped on many nails, couple rusty,
Uh-huh.
and every time my mom would take me immediately and pour just pure alcohol on
Ouch.
And
Yeah,
salt.
burns.
Sure.
Salt.
But-
too.
Yeah.
it. I
Notice
think she got rid of it.
Notice the shape here. We're going to see this little round. That's got one of those spores in there. So once again, the problem with Clostridium tetani is it's making a toxin. So I can kill the bacteria with air, you know, but the toxin will still be deadly. It causes every muscle to contract. So it's
That's
the opposite. This
much-
is the
That's
painful.
like
Everything contracts so much, and it just, and then your heart can't beat anymore. And you
That's
die.
an interesting painting.
I think that is painted by a famous painter, William Blake. I think Blake did that.
Imagine
So
if you
that
put both
guy's like-
viruses in at the same
He's
time.
about
I
to
wonder if
die.
they maybe
Yeah.
counteract each other.
Yeah.
I don't know.
So that
And they're
guy-
not viruses, they're bacteria, and it's a toxin.
Or bacteria,
That's
So that guy's like having the worst cramp ever.
it's terrible cramp. Yeah. So it's important for our muscles. If one is contracting, the other has to be relaxing. All right, Clostridium perfringens, and it's in the same group. Also an obligate anaerobe. It's down deep in the earth. It causes a disease. Now prepare yourselves. Although it's just a drawing, so it's not too bad. Called gas gangrene.
Ugh.
Yeah.
If I showed you a real picture, it's really gross. So when the guys in World War II were digging down deep trenches and then getting wounds, they would end up with this thing called trench foot, or trench foot was like a fungal infection, but if they got really bad, they might have in contact with Clostridium perfringens. And in this case, like this, it's called gas gangrene because when it goes through the process of actually growing in your tissues where the oxygen isn't, then it produces this gas that's real bubbly. And in this case, you have to amputate. That's the only way to kind of get rid of that. But it also is making a toxin, and that toxin can, even if this is dead, the toxin can still cause disease. All right, salmonella. You probably heard of this in relation to food poisoning.
Yep.
And so you can see it's in Grassula cutis. Scotobacterium is the genus species name of it. And what it does is you might intake it into your body, and it'll grow in your gut. It'll make you really sick. Symptom within 12 to 72 hours: nausea, vomiting, fever, diarrhea, abdominal cramps. 4 to 7 days illness ranges from mild to severe. Severe cases, infants and people who are immune deficient, elderly people, they can actually die from it. Bacteria travel your small intestine. They start their life cycle there. And sometimes they can get into your bloodstream, and if that happens.
Is this like sarsaparilla?
No, salmonella, no. This is, I guess the number one way of getting salmonella is through chicken products, eggs that are not cooked properly. So some people eat raw eggs, chicken that's not cooked. You know, we don't usually eat chicken medium rare.
Wait,
Right, we usually cook it.
So what's sarsaparilla?
Sarsaparilla is root beer.
Oh,
Yeah.
that's
So
not
root
at all
that
what
they
I
make
thought
root
it
beer
was.
out of. Yeah. So to avoid salmonella in your kitchen, this is interesting. Don't rinse raw chicken because it just will spread it. I thought that was interesting. Don't rinse it. You're going to cook your chicken to 165 degrees Fahrenheit. Wash your hands after touching chicken. We all know that, right? Always use soap in doing that, and then rinse really, really well. So try to keep salmonella in the kitchen. Escherichia coli, known as E. coli, is found in the normal flora of the human gut. It's in Grassula cutis Scotobacterium as well. There are strains that are pathogenic, and I want you to think for a second about ground beef. Okay, everybody likes a hamburger, yeah?
Oh yeah.
Brisket.
So when they talk about people who have been infected with E. coli that was of the strain that caused serious disease, serious enough to cause death in children before, and it's almost always a hamburger. And the hamburger hasn't been cooked thoroughly. So people don't like you to eat your hamburger medium rare.
It's so
I
good that way, though.
prefer my hamburger medium rare.
Yeah.
So if you know
I gotta
where
take
the
the
meat
risk.
came, so I want to talk to you about how ground beef is ground beef's the problem, not a steak.
No, this one has
So let's think about we're getting our meat from our cow, right? And we're in a facility and maybe it's not 100% clean or whatever. The E. coli comes from the feces, from the poop, all right? And that might get onto the meat. Now think about it. If I have a steak and I have bacteria growing on the surface of my steak, and then I cook my steak on the grill, the surface has all been cooked, right? So the bacteria is taken care of. But if I grind that meat up and I put that stuff into the center of that meat, do you see how it could still be present inside even after cooking? So that's why ground beef is the big problem when it comes to E. coli infection.
But it's fine eating it rare, right?
Say again?
It's fine eating it rare, right?
I feel like it is typically if you have a food poisoning from E. coli, it's not going to be a strain that's deadly, but every now and then there's a deadly strain that goes around. Okay, so we're going to talk about our lab time now, what we're going to do. I have these things here that we're going to be looking at called agar plates. These are Petri dishes with agar that I put into them last night. I poured them in there as a liquid, and I let them cool off, and now it's like Jell-O. This one's ugly, that's why I'm using it for me instead of sitting at home with you. Keep in mind, since I'm a microbiologist, this is my field, and I love this stuff, right? I love it. So I'm going to be sending something like this home, but before we do that, I'm going to show you how to treat it. I have a question. Does anybody have a pond? Can you bring some pond water next week?
I have a puddle.
In a jar?
Same.
Honestly, a puddle will work, but if you have a pond, maybe get a little, and we can look at it under the microscope next week. Okay, if you forget it, no big deal. I do have a culture I can grow, and I'll start growing it this week if I don't
I
forget.
can find a toad.
I could get some
I don't need a
really
toad.
nasty creek
I just
water.
need some water.
Some
Okay, can you
really
bring it in a
nasty
jar?
creek
I like
water.
I like
like
nasty. That's
really,
good.
really bad.
Okay, so culturing bacteria for study. Typically what we do in a microbiology laboratory is we try to get bacteria isolated so we can tell what we're looking at, because typically there's millions of them growing and we want to get little isolated colonies. That's our goal. So what we usually start with is a broth tube. It's basically soup in a tube, and we culture something by taking a swab or something like that. I have this really cool microbiology tool right here called a microbiological loop. In fact, I'll put it where y'all can see these. It's got a little loop on the end, and I use these to go and grab some bacteria off of something. Remember, there's millions there, so we gotta just get a few. All right, and then once we get it growing in the broth will go from clear to murky. You can see stuff growing in it. You may have seen this if you left something sitting out, a cup of lemonade, and all of a sudden it gets real murky. As time goes by, you're like, I'm not gonna drink that, right? You're gonna pour that out. It's probably bacteria growing in there. And then we'll take a little bit of that liquid and we'll put it onto what is called an agar slant. So we take this agar, the same thing that's in my petri dish here, it's like a Jello, and I'll put it, and I'll let it cool at a slant in a test tube. And then I'll take this loop and I'll put it into my soup that's got it growing right here. And then I'll just take a scoop of it and I'll streak it like this onto that slant. Now, why do you think it's slanted? What's an advantage there?
More room.
Surface area, exactly. So then I'll put it in a warm enough room. It's probably about 70 degrees. It's not that warm. It's what do you call incubator? And I'll let it grow, and it'll grow on there. And then I'll take it off the slant and I'll put it onto my plate. So I'll take a little bit of it and then I'm going to put it onto this plate. I'm going to have y'all do this here in just a minute, and I'll streak it onto my plate like this and then try to get an isolated colony from that. Okay, so then here we go. Agar slants can also have a needle, inoculating needle that sticks it down in. Now the advantage here is if I'm working with something that happens to be anaerobic, it'll grow down here and there's no air. On the surface it's going to have air, but it'll have no air down inside of it. So I might have some anaerobes growing there. Yeah, there's our increased surface area right there. All right. And then our streaking of our agar plates. This is kind of cool because whatever they're using, they put a blue light on it and it was glowing. I don't know that we'll have that, but I have a blue light I can put on there to see if it does glow. And we're going to see if we can find the different shapes of our bacteria. Okay, so colony formations, they look something like this. I'm going to send you home with an agar plate. I'll instruct you what I want you to do with it, and you're going to bring it back next week. Try to remember to bring it back next week, and we will actually probably try to pull off a bit of it, put it onto a slide and take a look at it, okay? I'll do this gram stain. I have my gram stain kit right here, and we'll try to do a gram stain. You will almost 100% have a contamination of mold in it because of how we're doing it. There's just no way to maintain aseptic technique. But you can see these are all little colonies. Look, you can tell they're different, aren't they? Some of them are different colors. Some of them have a different, this is called the margin, right here where it's not completely flat. It looks kind of scalloped on the edges there. We have the orange, the yellow. So we can have a circular form or irregular or filamentous or rhizoid. When you look at it from the side, you can see them standing up kind of like this. The margins can be like this, undulate. So that one here had undulations in it, you can see. All right, how to streak the plate. So what you're going to do, let me hand you a plate. Okay, I'll get this light on.
See the best streakers.
And I'm going to go ahead and stop the recording here.
Come on, that was funny.
Really quick, successful streak like this. So I'm going to go through all this really quickly. I'll put that up for the people who are not here to hear me. All right. And we're going to see we have to use aseptic technique in order to do this. And that's what we're going to do right now.